Good afternoon, everyone. We'd like to welcome you to our summer research seminar series for those that are joining for the first time. My name is Vicki Bowden, and I'm a project manager here in HOPP. Today, we have a great speaker. Before we begin and I do some introductory remarks, I would like to remind all of our attendees. So the first is that we do have a Q&A feature, and we ask that you submit your questions, and towards the 15 minutes of the lecture, we'll be moderating, and any questions that we can't answer, our speaker will answer them via Twitter. And for those that are joining after the seminar, you will be receiving a feedback survey, and we ask you all to complete that as well. And also, for those that haven't viewed other past recordings, we do have them available. You can visit our YouTube page, HOPP Summer Student Program, to view past lectures. And today, our talk will be the science of sequencing. And we have Dr. Caitlin Stewart, who is our guest speaker. Dr. Stewart received her PhD in molecular biology at the University of Cambridge Wellcome Trust Sanger Institute. She began her postdoctoral training here at MSK at the Center for Molecular Oncology, and she currently is serving as a postdoctoral research fellow in the Luis Diaz lab. She designs and optimizes sequencing panels. At the moment, she's working on a panel for clonal hematopoiesis of interdeterminate potential, so CH or CHIP. And it is a condition of the blood which predisposed to cardiovascular disease and acute myeloid leukemia. So this panel that she's working on will be used to monitor and study progression of CH in clinical CH clinic patients and to see if they would need additional treatment. Again, we'd like to welcome Dr. Stewart to our seminar.
Okay, so I'm here today to talk to you guys about the science of sequencing. I wanted to go over a little bit more about who am I and how did I get to MSK, as suggested by Ross Levine, a little bit about why I'm interested in sequencing and what my job is right now as well. And then I'm going to go over what is DNA sequencing, how does it work, why do we do it, and what are some examples of how it's used at Memorial Sloan Kettering. So I actually grew up around science and around academia. Both of my parents are scientists, and I grew up basically in the lab and in the hospital following them around. I absolutely loved research and science from the beginning, and I had a good idea of what it was like to have a career in science. So this is what I've always wanted to do, and I'm very happy to be doing it now. And I think it's unusual, definitely, to be so narrow-minded right at the beginning, to have such a strong idea of what you want to do. So don't worry if you don't yet. I moved from Canada to the UK to do my undergraduate degree. And I worked in a lot of different science institutions as I was doing my undergraduate. So basically every summer I spent in a lab somewhere. The first one was with my dad's company working back in Canada, and I was working on drug purification for anti-botulinum toxin drugs, so sort of Botox related drugs. And then I spent the second half of that summer working with a group called Operation Wallacea in a place called Utila in Honduras. And I was helping other PhD students sort of fulfill their research in marine biology. So looking at juvenile fish populations, seagrass density, and then also doing a project on my own looking at scuba diver damage to reefs. Even if you're trying to be quite safe, people kick stuff by accident. And we were trying to work with the local community to figure out, should we have only a certain part of the reefs open at certain parts of the year and allow for it to recover at different points. I really enjoyed this, and I love marine biology, but for my second summer, I decided to go to the Bermuda Institute of Ocean Sciences, and that's where I got interested in molecular biology. I started working on RNA expressions in actually sea urchins. So sea urchins, the ones that you eat and most people think of are very small, sort of round. But in Canada, And off the coast, they can grow up to several feet wide. They live very, very deep in the ocean. And the big ones live for hundreds of years, and they have low cancer rates. So we were trying to figure out how something so big and that lives for so long doesn't sort of develop cancer. These type of sea urchins actually have active telomerase and certain types of RNAs that are expressed that you see in certain types of cancers as well. So telomerase is usually expressed in embryos, and it allows the cells to repair their chromosomes so that they don't break down and then the cell dies. So this is what I was studying there, and I started getting interested in molecular biology and how it relates to cancer and other aspects of what could be studied. I decided to do more of like an actual lab rotation rather than working just in the ocean for the following year. And I worked at the Institute for Cancer Research in Edinburgh. There we worked on a gene called EIF1A, elongation initiation factor 1A, in cells and in mice. And in the mice, when you overexpress the gene, they develop certain types of cancer. And when you underexpress it, they have neurodegeneration and problems with walking and stuff like that, which is pretty interesting how one gene can cause two different types of conditions. I then did my honors project. So in Scotland, it's a 4-year bachelor's with a year where you take specialist classes at the end and a 6-month sort of longer research project. So you're in the lab 6 months full time and you write a small thesis. And when I was there, I worked for Professor Nick Hastie, who was working on Wilms tumor, which is a type of cancer in children in the kidney. And I was working on a gene called beta-catenin and how it signals in mouse embryos and mouse cell lines as well. During this final year, I really loved molecular genetics and biology, and I started taking courses in genomics and in gene editing, just two of the things I'm really fascinated by. During my course in genomics, I found out at a place called the Wellcome Trust Sanger Institute, which is located in Cambridge and part of the University of Cambridge network. The program is a bit different to a typical PhD program. It's called a 3+1 program. And this appealed to me because I didn't actually do a master's between my undergrad and my PhD. I just went straight to studying the next thing. And in a 3+1 program, you do 3-month rotations and then pick the lab for your PhD. So this is useful because you can get a sense of the lab culture, you can learn new techniques, you can work on a variety of different things, and really try out some stuff that you've never done before. At the Sanger Institute, they mainly focus on genomics and molecular biology, but they also had the largest mouse house in Europe at the time. So they were a big model organism research center as well. And they really focused on you doing both wet lab and dry lab or computational rotations. So all of the computational students had to do at least one lab rotation, and all of the people like me in the wet lab had to do one set of coding. And I had never opened a terminal or done any coding before, but I found this a really rewarding experience. And I suggest to everyone, if you want to be a wet lab biologist, you should definitely go ahead and try and learn a bit of coding, at least a little. So my first rotation, I was working on cell culture techniques in zebrafish. The type of editing that you do in mice doesn't actually work in zebrafish. This project didn't go that great. My reagents arrived at the end of the week, like in the last week of my actual rotation. But I learned a lot of different techniques and working with people in the lab, and I really liked the lab culture, and that's the lab I ended up actually joining for my PhD, even though that particular project didn't work very well. My second project, I was working on the design of zinc finger nucleases, and these are a type of protein that you can use to edit the genomes of cells. This was before CRISPR existed, and this is what we did for a little bit, maybe a year or two before then. And then in my third rotation, which was my computational one, I worked on the Tasmanian devil facial tumor disease, so Tasmanian devil cancer, and they spread it mainly by biting each other, which is how they say hello. And I identified different types of translocations and then confirmed them in the lab as well. And this was, as I said, hard, but really worthwhile to learn how to do. In my PhD, I actually moved more to studying developmental biology as opposed to cancer, and I studied RNA expression in oocytes and developing embryos, not just in zebrafish, but in xenopus and mice as well. And then when we found interesting genes involved, I knocked them out using CRISPR, since CRISPR had just been invented the year before. It was really interesting, and my main interest, similar to the sea urchins in early development, has to do with the fact that a lot of these programs and in terms of transcription, all of that is repeated in types of cancer. So the same things that drive proliferation in an early embryo also drive proliferation in certain types of cancer. And I find that crossover very interesting, how it's able to be stopped in embryos, but not in types of cancer. While I was working on this project, I actually discovered a new type of small RNA and developed some sequencing methods in order to be able to sequence it and find it. And I really enjoyed this aspect of, developing sequencing technologies and learning and trying new sequencing methods. And that's what I decided to focus on for my postdoc when I joined MSK. So I first joined the Center for Molecular Oncology back in 2016. And then I was originally working on cell-free DNA sequencing, which is a non-invasive way of detecting cancer. And I'm going to talk about that a little bit later in the talk. And then following my love of RNA sequencing, I was also trying to develop methods for cell-free RNA and looking at expression profiling. Now, as of January, I've joined Luis Diaz's lab, and I'm developing a panel for clonal hematopoiesis. And then also ones for cell-free DNA, and we're trying to look at their underlying biology. And I hope to do some cell-free DNA sequencing soon, single-cell sequencing soon, to try and look at the function of each of the individual cells. This, for me, has actually been less academic and more of an industry position. So I work with a lot of different companies looking at turnaround time, cost, what it would take for them to implement the assay as well, and how we can scale up. So it's not just me doing experiments. I've got a collaborator who wants to run 1,000 samples, and me doing that by myself in the lab is very difficult. So it's been a lot more about, how to scale these things up, and then also working with multiple different researchers to see, how can I help them achieve their goals for research. And I've been really enjoying this aspect of the job so far. It's a bit different from typical academic research, but it's been really good. So just to go back to the beginning of what I was going to talk about for the whole lecture is what is DNA sequencing? How does it work? And why are we doing it? So DNA sequencing is done for a variety of reasons. For instance, you can look in terms of research at what genes are there, try to look at the evolution between different species, or even what's present in, different individuals. So from one person to another, or from one tissue to another. In terms of looking at cancer versus normal tissues. And then for clinical care, we can use sequencing as well. So you find mutations and different types of variations, so copy number or translocations as well. And we can try and look at predisposition to disease. So the sequencing that I'll be talking about today is about sequencing the whole genome, or the entirety of certain genes. This is quite different to what you see in forensics or things like 23andMe and ancestry testing kits, where they focus on only looking at very variable parts of the genome, whether or not they're within genes that are important for disease, or just in genes in general. So these are called, they're called SNP arrays, and they're different from what we're looking at today. So sequencing is so important in cancer genomics because cancer is really a genetic disease. Cancer results in changes to the DNA and changes that can be inherited through different generations. And I don't mean generations between, germline predisposition, but really somatic changes or changes due to carcinogens or exposure like UV or tobacco, or maybe there's errors in the DNA of the cell that causes errors in the replication. So these are somatic changes that only affect some cells and that end up being cancerous. There are also, as I mentioned, germline variants that predispose to certain types of cancers. And we study those as well at MSK. You can look at both together, but the main part of what we look at, because most cancers are somatic, are the somatic variants that are, or sporadic, are the somatic variants that are gained. So just to go over a couple definitions of words that I've used already, and we'll keep using quite a lot. Germline is something that's inherited. You can get it from one or both of your parents. There's a somatic variant, which is something that's non-inherited by carcinogens or maybe random processes in the cell. I use the word variant instead of mutation, and this is a more all-encompassing term that could be a mutation, it could be something different in the copy number, or it could imply a type of translocation. So copy number is a region of the genome that is amplified or deleted. So you have extra copies of genes, or maybe you're missing a copy of a gene, and that affects the expression and can drive tumorigenesis. A translocation is where two different chromosomes or one chromosome is broken apart and fused back together again in a different orientation. So this can cause things like fusion genes, where two genes have been brought together and maybe the expression changes between what they normally would be, or the trafficking within the cell changes. So maybe the location the protein goes to is different. An example of this is something that was mentioned, I think, to you guys before, which is the BCR-ABL gene, which causes types of leukemia. So the original sequencing methods that were developed back in 1977 were called Sanger sequencing. This was developed by Frederick Sanger, who the Sanger Institute is also named after. It was used in the Human Genome Project, which is how we have the human reference sequence, and that took 13 years and cost $2.7 billion. This reference genome sequence is still maintained and is available to anyone for free. I think this was really important in terms of not patenting, the information and making it available for researchers. But it was really big and it took a long time. So the human genome is about 3.2 billion base pairs. And to think about how long it took for them to make that, the Sanger sequencing is slow and you only do about 500 to 700 base pairs at a time. And then you want those sections to overlap so that you can look and piece together how the puzzle, builds a whole genome again. So this is briefly how Sanger sequencing works. This just shows some DNA replication. So you have the template DNA that you're interested in sequencing, and then you have a primer and then a new DNA strand. And this is how DNA is replicated as well. So you have DNA polymerase that adds the nucleotides on. And the trick with Sanger sequencing is that instead of regular nucleotides, we use fluorescently labeled nucleotides. So for each sequence of DNA, we have a fluorescently labeled nucleotide at the end of it. The shortest ones will flow through a capillary first because they're shorter, and the longer ones come through at the end. As you flow these through the capillary, the ones at the bottom get excited by a laser first, and then you count and mark them one at a time as they go through. So the first ones coming through the excitation laser and detector are they're an adenine base, and then it's put out as detected as flashes of light onto the trace that you can see on the right side. And then this is converted back into the letters of the sequence of the DNA. So A, T, G, C, etc. If you look at how sequencing has changed over the time, I said the Human Genome Project began back in 1996, and it was first discussed in 1984. If it costs $3.2 billion, how do we have sequencing now that's available for research or for looking at whole genomes of patients? And you can see that there was quite a fast gain in terms of the number of different genomes that have been sequenced for different species. And now we have several tens of thousands of different organisms sequenced, and the ability to use whole patient genomes for human healthcare. And that's because of the change in technologies that have become available. So I mentioned the first one was Sanger sequencing, which you can see on the far left. And then the next one I'm going to talk about is second generation sequencing or next generation sequencing. And then there have been some third generation sequencers which have been made now, which have longer reads, but slightly higher error. So they haven't been as widely adopted as types of technology yet. The main workhorse of what everyone uses for DNA sequencing is the Illumina machine, which you can see in the middle, which was developed in 2006. So next generation sequencing, as I said, Illumina is the most common. It works a little bit similarly to Sanger, except instead of flowing through a capillary and taking pictures with the laser, it uses clusters of all of the sequences together and then takes a photo across a whole flow cell. And so in this way, you multiplex the reactions so you can do millions at the same time. Although the length of it is shorter, only 150 base pairs. So you have, millions of data points and then across 150 times for each of those. So computationally, the amount of data that you actually have there is huge compared to Sanger sequencing. But being able to multiplex all of these things and work on them at the same time means that overall, it's a lot cheaper to use. So this is how the cost of the human genome has come down. Back in 2001, it probably cost $100 million for someone to sequence a single whole genome. By the time I did my PhD in 2010, it was around $50,000 per person for a whole genome. And then if you look now or last year in 2019, it's around $1,000 a person. There are some companies, there's one called BGI in China that say that they've done a $100 genome. But we still have to see whether or not it's actually $100. But that would make it amazing for being able to do personalized medicine and able to offer this to more patients than we already do. So as I mentioned, bioinformatics and computational biology, especially for sequencing, is incredibly important. So the amount of data that you have is massive. I've shown an example there from the Wellcome Trust collection, which shows a printout of a whole human genome. So each page is just ATGC of what the sequence is of that chromosome. And you can see it fills a whole bookcase. So most of this data is actually stored on Unix-based computational clusters of data, and it won't fit like on your laptop. Me alone in our small lab, where there's only a handful of us doing this type of sequencing, we have 15 terabytes of storage, and we're, as I said, a pretty small lab to be doing this. So if you're going to be doing huge sequencing projects, you need a lot of space. So this is where I'm going to plug again that you should try and learn how to code, at least figure out how you can access your own data, even if you're in the wet lab. I found it really helpful in terms of being able to explain what I want in terms that maybe the computational biologists understand better. And I've been able to, understand a lot more in terms of what's actually possible with the data as well. But if you work in computational biology, you can either work in just processing the data or also doing things like writing your own computational programs to, analyze or process the data as it comes off of the machines. And there's a lot of opportunities in that area. So here's some terms again, that'll help make understanding sequencing and the technology of next generation sequencing a little bit easier to understand. So a DNA fragment is what we call a small piece of DNA that makes up a sequencing library. So this is when you take genomic DNA and you break it into smaller fragments. And so these fragments are about 150 to 200 base pairs. A sequencing read is the computational version of that. So it'll be the actual sequences as a string. So like TTACTA as I've written there. And a sequencing library is when you have those DNA fragments and you've attached adapters on the end. An adapter is a type of PCR primer with complementary sequences to what's on the flow cell. And it will allow you to attach the library and actually read it. We say sort of like as if it's a book. And then the flow cell is just a part of the sequencer where you not only attach the library, but this is where you flow over those fluorescently labeled nucleotides, and you flow them over the flow cell, allowing them to bind each to the strings of DNA. So this is what that looks like in terms of prepping your own library. You have your genomic DNA and you fragment it to get the DNA fragments. You can do some targeting, and I'll talk about that in a little while. But to make it a DNA library, you just attach the adapters and set that up for your sequencing. This is what a flow cell looks like. They can be quite small. The ones for the MySeq that I have are about the size of a battery. And you can see what the clusters look like in that lower image. So each DNA fragment is attached to one of those adapters into a location on the flow cell. And then you do some PCR to amplify them so they form a cluster. You could see the clusters in step 3 of the second part there. And this allows sequencing up in one direction and then sequencing down in the other direction across a bridge. So that's what the NextSeq, the third image, is an example of an Illumina sequencer. And then you can see those images of the clusters again. So this is what it looks like after you've flowed the fluorescent nucleotides on. And each cluster itself will all be adding the same base at the same time. So it'll look like a very small dot, and the dot will be different colors. So the computer then figures out the order of each of those dots and tries to determine if each dot is sort of separate. And that's sort of, if they're too close together or too far apart, it might be difficult for the machine to read. So there's a sweet spot where you got to get it. And here it then takes each of those sequences from the different colors and converts it into a sequencing read. So this is what that kind of looks like. You have your genomic DNA, and then you break it up into all of these sequencing reads. And then what you have to do is put the puzzle back together. So you have to align them. And what we typically do is take the reference genome that was made during the Human Genome Project and then compare to all of the different sequences that we currently have in our sequencing reads. And here they've shown it with colors for each of the different sections, but yeah, so you align them back together, and this is done computationally. So just to go over that one more time, the images are converted into text files on the machine, and then you do some QC to check for errors, make sure the clusters weren't too close together. You do alignment of the reads to the genome. So I've shown that at the bottom here again. You can see, say the blue is your sequencing read, and you've got the A, T, C, T, T, G, T, A, G, G. And then, so you want to read along your reference genome until you find that location. And once you do, where it's been placed there, you create a new file that has not just the sequence of the DNA that you've just sequenced in the sequencing read, but also the position of where it's located. And you've put this into a type of file called a binary alignment mapping file, or a BAM file. As a binary file, it's easier to store, it's much smaller. And then you run additional programs trying to look for what you're looking for within the gene. So are you sequencing to find mutations, or different types of genes that are present if it's a different organism, things like that. And then you can output the files and images of what you're looking for. So if it's just the list of mutations, you can just get a text file out that says that. There's two main things to consider when you're sequencing something, the depth and breadth. So sequencing depth matters because you want to have a certain amount of confidence in what you're looking at, and higher amounts of reads in that area means higher confidence in what you're looking at. So you count the coverage and the depth at a certain point by looking at the number of reads at each position. So if you go along from left to right here, there's 5 of those blue reads stacked on the reference genome, so the coverage is 5. Sometimes the coverage is 2 or even 0. If you're looking for a mutation that you know is there 50% of the time, you might not see it out of those 2 reads, especially if you have 0 reads or only 1 read. So you want to have multiple copies in the same location. For germline and whole genome sequencing analysis, we usually look for about 30 times coverage, or 30x coverage. So instead of 5, it would be about 30 in that location. But if you're looking for mutations, you want to have higher sensitivity. So we do about 500x coverage in each location. So the problem with doing that much coverage for a whole genome sequencing library is it's incredibly expensive. If it costs about $1,000 to do a 30x whole genome sequencing library, you can imagine that 500 times is too expensive to do for patients. So what we do instead is something called a targeted panel. And this is where you select just the genes that you want to sequence and sort of throw out everything else. And to capture them, we use something called probes with complementary molecules that are complementary molecules to what you want to pull out. So it's either DNA or RNA that is complementary to your gene of interest. And then they have chemical tags on them. And to do this, you can make different size capture panels and change the genes that are involved. So I put 2 examples along with the whole genome sequencing down here. So one of the biggest ones, if you want to look at all of the coding genes in the genome, or maybe all of the exons, you can do exome sequencing. And this is where you look at only the exonic sequences. It's quite a large sequencing panel, and because it's still quite big, we typically only sequence to around 100x. And something we've developed here at MSK is called MSK Impact, and this is sequencing only the cancer-related genes so that you can focus your interest in. And this is sort of a medium-sized panel, and we do these at about 700x coverage. So targeted capture works by developing these probes or baits that I mentioned before, and they're attached to a chemical and a molecule called biotin. And then you take magnetic beads, which have something that are coated in streptavidin. And streptavidin and biotin bind together really strongly. It's very difficult to break them apart. So what you do is allow your probes to hybridize, and the complementary sequences will match what you have in your sequencing library. You add in the streptavidin beads and then wash away everything else by using magnets. So this is a quick overview of what that looks like. So say you have your magnetic beads, and in green are your biotin labeled probes and your DNA of interest bound together. You put them in, and the biotin and the streptavidin magnets will bind together really strongly and specifically. And then what you do is you put a magnet next to your tube, and that will bring all of the magnetic beads and your DNA of interest along with the biotin probes. And you can wash away everything else in part B. So you remove all of the liquid, and then you can add new liquid, which allows the probes and the DNA of interest to separate. And then you're left with only the genes that you want to sequence. And this is how we do a targeted capture for making libraries from smaller amounts of genes. So MSK Impact, as I said, is an example of this. And MSK Impact is approved for clinical sequencing, so we can do it for patients and give them back information about their tumors. It's at the moment 468 cancer genes. And when I checked yesterday, we had over 57,000 samples up on CBio portal. We do about 10,000 sequences per year. Mostly those are different patients, but sometimes a patient will get sequenced twice if they have a metastasis or just to recheck the tumor if they have a recurrence. So all of this data and sequencing data is available on CBio portal. I'd recommend you go and have a look at it. You can click on different subtypes of cancer or different types of studies, and then look at which mutations are the most common, or look at survival data for each of those. What we do with MSK Impact mostly is to look at a patient's cancer or metastasis and try and ask questions about it. So are there mutational patterns across that type of cancer, or does having this mutation for this patient mean that they'll have a better or worse prognosis? If a patient presents with 2 cancers, are these cancers related, or are they not? Sometimes they're not. Sometimes people are unlucky. And most importantly, I think one of the things that we do at MSK is looking for drugs that we can prescribe based off of these mutations. So to do this, we do something called a basket trial. And basket trials are different from normal clinical trials, where a clinical trial, you would say that you're going to target a certain patient population and then maybe look at the mutations and stratify them. Whereas a basket trial, you don't care about what subtype of cancer they have. You mainly care about the mutation and whether or not the drug that's been developed that targets that mutation is good for that individual or not. So there's a drug, the first one that was a basket trial that was done at MSK was with a drug called vemurafenib, and it targets a mutation in the BRAF gene V600. This is a drug that was very effective in melanoma, where BRAF mutations are very common, but there's a lot of other cancer types that showed, the same mutation, and we wanted to see whether or not this would work. So in the infographic below, they have different patients, each with a different type of cancer, and the patients with the dots in them are patients that have that particular mutation. And so they chose all of those patients and put them in one basket to test them together, which is why it's called a basket trial. This is an example of how the vemurafenib trial was set up. You can see all of the different cancer types and then aspects of what they were looking for. So you can see everything from non-small cell lung cancer, breast cancer, ovarian, even multiple myeloma, which is a hematological cancer, and colorectal cancer are in there. So they took a set of patients and tested them all with the drug. And these are some of the study results. So these are a type of plots called waterfall plots. And what you do to read them is at the beginning of treatment, they take a scan and look at the size of the tumor. And then the plot shows the change from 0 or the baseline until the end of treatment. So any bar that increases above 0 is an increase in tumor size, and any bar that goes below is a decrease in tumor size. And they have significance cutoff points shown in the dotted line. So you can see in panel A for non-small cell lung cancer, the patients did really well. There were only a few patients who did poorly on this therapy. The same was seen for colorectal cancer, although it was about a 50-50 split in terms of who did well. And the smaller other groups, like glioma, was a little bit more variable. But overall, they found that, this drug works in other patients, and maybe we need to be expanding this type of study. This is what David Hyman's been doing at MSK for several years now, is more basket trials to see if we can expand the use of existing drugs to other patient populations. One of the other things I think is really interesting that we can do with MSK Impact is looking at tumor mutational burden. So mutational burden just refers to how many mutations a particular patient has. And we usually talk about this in terms of mutations per megabase, but you can also look at the number of mutations in the total in the tumor. So things like AML, acute myeloid leukemia, you'll have maybe like 1 to 10 mutations or maybe more per cancer, whereas things on the other hand, like melanoma, you can have upwards of 1,000 or 10,000 mutations within the cells. This is important because a TMB is related to your response to immunotherapy, which is another type of therapy that we do at MSK. So immunotherapy, I think other people are going to talk about this in more detail, but it's a type of therapy where you train your immune system to recognize types of cancer cells. And in this case, the more mutations you have, the more unlike yourself the cancer seems to be, and the more easily recognized by the immune system. So in this case, they separated into the top 10% highest TMB, the top 20 to 10% TMB, and the bottom 80. You can see in this overall survival chart, the patients that do the best, the top is the top line and blue, where the more mutations you have, the more benefit you have from immunotherapy. So there's tons of other types of sequencing. As I mentioned, I really like looking into RNA-seq, but you can also look at epigenetics or relations to proteins. So for RNA-seq, you convert the DNA into cDNA or complementary DNA using reverse transcriptase enzymes, and then you can see what's expressed by each of the genes in different cells. Methylation you can treat with a compound called bisulfite, and this will allow you to mark the DNA about where the methylation locations are, and then you can look at methylation or chromatin marks. The ChIP-seq is chromatin immunoprecipitation, and this is where you bind DNA and protein complexes together and then release them and sequence the DNA that you have remaining. And it'll tell you about the location of different things. This is useful for histone modifications or histone binding sites and transcription factors. So where are these genes and where are these proteins interacting with the DNA, and how is that affecting things like expression or maybe functionally as well. I'm not going to go into too much detail about the different types because there's probably about 100. The graph here shows over time from 2006 until 2014, but there have been many more developed since then, and each bubble is a different type of sequencing. The size of the bubble relates to how often and how many citations it has and how much it's been used. So you can see RNA-seq is a very popular technique, and others such as ChIP-seq are also in there, but a little bit smaller. There's, yes, tons of different things you can look at and develop. One thing to be aware of in all types of DNA sequencing or RNA sequencing or anything is the aspect of tumor heterogeneity. So when we do sequencing, we take a biopsy, and a biopsy is normally with a small needle or a small section of the tumor taken during surgery. And different areas of the tumor actually have different cells and different mutations in them, and they may not be representative. So if you look at the image at the bottom, you might have the start of a cancer, which is one cell with one red mutation. And as this cell divides, maybe one of those red cells gets another mutation, say the purple one, and then you've got the red and the purple there. And here, if you sampled it, all of the cells would have both the red and some would have the purple. And you can see that in the third as well. But if one of the red cells then also mutates and gains another mutation, but it gains a different mutation from the purple, say the blue one, there you can see that the position and that there's differences in the cells that are available. So some will have red and blue, and some will have purple and red, and some will just have the red. So depending on where you take your biopsy, you might miss the fact that there are some blue mutations in there. And this is important for determining what drugs patients get or understanding how a cancer is driven. And it could result in the patient having another recurrence because you haven't double checked all of these things. So what can you do? One of the first options is to sequence multiple areas of the tissue, take different samples. Another option is to do similar to that, which is single cell sequencing. And then a third option that I'm going to talk about is cell-free DNA sequencing as well. So single cell sequencing, this is an example of single cell RNA sequencing, is just sort of how it sounds. You separate the tumor tissue into a whole bunch of different cells, and then for each cell, you sequence it separately. So when you do bulk sequencing, you lyse all the cells, and then they release all of their RNA or DNA. And you sequence them together, and then you get sort of an average expression or an average profile from each of those cells. You don't understand what the differences are, or what the subpopulations could look like. But when you do single cell sequencing, you get a specific expression profile for each of these, and then you can look at the subpopulations individually. So this is useful not just for looking at the mutations or expression in different cell types, but also looking, for instance, at immune infiltration. How many immune cells are there? What type of immune cells? That can be very informative in terms of what type of cancer it is, or the prognosis. As I said, you can do single cell DNA sequencing as well. So here you have an example of 5 different cells that you've sequenced, A, B, C, and D. If you sequence all of these together, you would see all of the mutations at once at different frequencies. But when you sequence them separately, you can see that all of the cells actually have this one blue mutation. So this is probably a clonal mutation that was the first one, like the red one in the previous slides. And then you can see there's sort of a split where some have yellow and some have red. And this allows you to then work back and draw a sort of lineage tracing tree, like a phylogenetic tree that you can see on the right. Now, I've added the letters for where each of those cells probably originate from this tree-like design. You can see how the mutations are gained sequentially. This is a slide that I stole from Ross Levine, where he shows exactly this applied to his work in AML. So I think the size of the dots here represent how many of those they found in their single cell data. And at each step, like the one on the left, you can see that first the cells gain a DNMT3A mutation, and then an IDH2 mutation, and then finally an NPM1. And this is an example of what happens in sort of more of a straightforward, all the cells are clonal within that patient, gaining mutations sequentially. But what can happen on the right-hand side is, for instance, a patient has an IDH1 mutation, and then some of the cells, like the purple and the blue, develop different mutations. So one has the FLT3 and one has NPM1. And then what happens in the FLT3 mutation is that one actually also then gains a different NPM1 mutation. And this you wouldn't have been able to see the differences in these trajectories if you'd sequenced everything together as bulk sequencing. And this is one of the important and really interesting things about doing single cell DNA sequencing. The last thing I want to talk about is cell-free DNA, which is something that I've been working on during my postdoc. So cell-free DNA is actually released from all your tissues as they die and turn over into the bloodstream. And this DNA can be sequenced to find the tissue of origin. It's used quite often in fetal maternal medicine to find out if the baby has any aneuploidies like trisomy 21. So you can actually do this without having to do amniocentesis, and you do it just by taking a vial of the mother's blood. It can also be used in different aspects, looking for like damage due to trauma or in sepsis or infection. But we use it here to look at circulating DNA coming from tumors. These are present at a low level in the cells. In the plasma. You can look at different aspects, but what we mainly focus on are the point mutations, copy number alterations, and rearrangements. But you can also look for methylation or focus on things that are within, say, apoptotic bodies or exosomal DNA. At MSK, we use something called MSK Access, which is another targeted sequencing panel. It's only 127 genes, so it's quite small. And we use it to look at the circulating tumor-derived DNA. We sequence this to about 10,000x, so very deep because it's a small panel. This is useful if you can't get a biopsy or as I said, you're looking for tumor heterogeneity, because as the cells die, they all release a little bit of DNA into the blood. So you can hopefully find stuff that you wouldn't have been able to see otherwise just by doing a single biopsy, or if you can't reach the tumor to do a biopsy. Then you can also do studies, because it's so easy just to take a blood draw, compared to biopsying a patient longitudinally while they receive treatment, or to try and find an early type of recurrence, or if they have resistance mechanisms that develop. So different organs and different types of cancer both release different amounts of cell-free DNA. It has to do with not only the tissue itself, but some aspects of where it's located. Here you can see like bladder and colorectal cancer, 100% of the patients we are able to detect the ctDNA in. But for something like glioma, it's a very small percentage. And we actually use cerebrospinal fluid instead of blood to look for these. We think it might be related to the blood-brain barrier, stopping the cell-free DNA from being able to access the bloodstream properly. So not only does the tissue type matter, but the size of the tumor and how active it is. So higher stage tumors, I believe this is for colorectal cancer, release more cell-free DNA than early stage tumors. And that makes sense based on the number of cells that are present. But also it's quite reactive, and the dynamics of cell-free DNA are quite fast. So if you do surgery and then remove it, within just a few hours, all of the ctDNA within the patient's blood will have disappeared. And you can actually use this to look at whether or not the patient's tumor will recur after surgery. So here's an example of one of those studies where after surgery, they took samples from patients, and in patients where they saw ctDNA again, the patients did poorly. And this might be due to maybe the tumor wasn't completely removed, or there are additional cells left over that are microscopic, or maybe metastatic sites that they hadn't found before, versus patients where ctDNA was not detected, and those patients did much better. Since you can detect it after surgery and at very low levels, what we've been developing now is ways of early screening for cancer. And this is an example of how they've done that in Hong Kong. So nasopharyngeal cancer is associated with EBV in Hong Kong. And Dennis Lowe and colleagues screened 20,000 individuals looking for EBV cell-free DNA in blood plasma. If those patients turned out positive, they came back for a second test. Some of them might have just had Epstein-Barr virus as a part of having mono or glandular fever. But if it was persistently positive and positive on a second test, they were sent for an endoscopy. Of those 20,000 patients, they did find some people with nasopharyngeal cancer, and you can see that the distribution of patients differed from those who wait until their symptoms in order to find them. So the panel A at the top, you can see a greater proportion in stage 1 and stage 2 were found than patients that normally come to the clinic, which have stage 3 or stage 4. Similarly, in terms of progression-free survival, the patients where they found the cancer earlier, they were able to get treatment earlier, and their overall survival was better. So that's some examples of sequencing at MSK. Thanks so much for listening. I've got just one more thing to leave with you, and that is that science is mostly failure. You need to learn from what didn't work. Don't take things personally. If an experiment fails, it's not you failing. Things just don't work sometimes. And it's okay to make mistakes. Just keep trying, keep working on what you're working at. Yeah. Okay, questions?
Great. Thank you so much, Dr. Stewart. So we do have a couple of questions. And as a reminder, any questions that we can't answer here, please submit them on the Google form, and Dr. Stewart will answer them. So Pravalilka asks, how many samples do you sequence in a typical day?
I have a very small sequencer in the lab. And I don't work in the core facility. So me, myself, I only do about 20 a week. But for MSK Impact, we do about 10,000 a year. And cell-free DNA is about another 10,000 a year as well. So there's a lot of sequencing that happens.
Right. And Paul Olapade asks, which coding language is really important for someone looking to do research in molecular biology or oncology?
There's so many that are used by different people. When I was learning, the most popular language was Perl, but that seems to have been fallen out of favor at the moment. Most people seem to do Python. I know a little bit of R for making graphs, and I quite like that. But yeah, I think I'd recommend Python as a place to start, or just learning Unix first.
Okay, so we've gotten a couple of questions regarding CRISPR. Could you provide more context, like what CRISPR is?
Oh yeah, sure. So CRISPR is something that was discovered in, I believe, a type of bacteria, and it's a way of fighting invasive pieces of DNA that come into the system. So CRISPR is actually a combination of a protein that binds to the DNA and a guide RNA that's produced by originally the bacteria, but now we produce them in the lab. And the guide RNA allows you to bind a specific region of the genome that you're looking for, and then the CRISPR protein will come, the Cas9 protein it's called, and it will break the DNA in that specific location and create a double-strand break. So what we've been doing in the lab is changing them so that we can create a break and then put in a sequence that you want, or then alter the sequence within that organism. And this allows you to make more precise changes. There are some people investigating CRISPR and using this for things like gene therapy, where you want to go in and specifically change a location within the genome and correct it, maybe say for cystic fibrosis or something like that. It's a very cool gene editing technique.
Okay. Paris Robinson asks, does studying common sequences of different organisms benefit doctors when figuring out if a patient has cancer?
I think understanding the evolution and how different things can, proteins have been made and stuff can help. Basic biology and researching that kind of stuff always does relate back to human disease and how we can use it. I mean, even when I was studying oocytes, the thought was like, it could be for fertility, it could be for cancer studies, it could be for a variety of different things. So I think so, yes.
Okay, great. Anish Bachu asks, what's the benefit of using cell-free sequencing over single-cell sequencing?
Single cell sequencing is very expensive. The experiments cost around $10,000 each at the moment. Cell-free DNA you can do for our MSK Access assay is, I think, several $100s or under $1,000. And it's easier to analyze the data.
Okay, great. We have an anonymous attendee. They ask, concerning cell-free DNA sequencing, does the cell-free DNA come from cells after apoptosis or necrosis?
We're still figuring that out. Mostly it seems like apoptosis. And the fragment sizes that you find are very small, around 150 base pairs, and they're associated with what you see when you induce apoptosis in cell lines and things like that. So we believe it's apoptosis mainly, but people are still working to figure it out.
Okay, great. So it's a good thing you said apoptosis, because the follow-up question, if it is, if the DNA, if the cells themselves underwent apoptosis, does that mean that something went wrong within the DNA to make the cell, quote unquote, kill itself, per se?
Not necessarily. I mean, skin cells die all the time and they're replaced. It's the same thing in certain types of cancers. Just all of your organs are replaced by new cells sometimes. It can be because of drugs and other treatments. We do see a slight spike in cell-free DNA after giving a treatment like that, where all of the cells apoptose and die. But it's not, it might just be the natural end of the cell's life.
Okay, great. We have a question more so specific to your work, and as a researcher, Joanna Lee asks, what's the favorite part of the work that you do, your job?
I think the people that I work with, it's been, for me, really important to be with a lab and people that I like spending time with, because I spend so much time there. And when you get to discover something new and you're still the only person that knows that thing, that's one of my favorites.
Okay, great. And another attendee asks, could cell sequencing benefit in combating COVID-19?
Yeah, the types of single cell sequencing. Do you mean cell-free or single cell?
They didn't specify. They just said cell sequencing in general.
Yeah, so we're looking into cell-free DNA sequencing from saliva from COVID-19 patients, and it's something we're working on at the moment, but also looking at things like single cell sequencing, or looking at the evolution of the different viruses by looking at in each individual person and rebuilding those phylogenetic trees is something that's happening as well. Yeah, there's a lot of sequencing that's being done in this area. Yeah.
Okay, great. And another question, is it true that the smaller the panel, the larger the coverage?
You can do whatever coverage and depth you want, but it's a cost benefit sort of analysis. So the smaller the panel, the deeper you can go for the same cost. If you want to do a cheaper one that's a smaller panel with less depth, that's available. Yeah.
Okay, great. So Siddharth Raghavan asks, how can scientists get around the fact that there is a lot of non-coding DNA in the genome when they are trying to map it?
So we use something called a masked genome, and that removes a lot of those repetitive sequences. It is a big problem, and it's something that's written into the alignment algorithms of trying to figure out regions that look similar or regions that are very difficult to align to, especially sequences that are, we call them AT-rich, that have a lot of A and T bases. It's very difficult to map in that region and to align there. It's an ongoing problem.
Okay, great. And another question we had is, would you suggest majoring in, what do you suggest majoring in undergrad? And I'll actually develop that question because we've gotten a lot of questions regarding like, what did you study specifically? So since our target audience are high school students, what would you recommend that they do now in preparation if they are interested in a career such as yours?
Yeah, as I said, take a coding course, study biology. Physiology is helpful to have as well. The university systems in the UK where I went are very different to how they are in the US. You don't have general years of study beforehand. You go straight into studying biology if that's what you want to do, which is why the final years at the end are much more specialized in terms of the courses that you take, and you do these research projects. And the PhD structure is different as well. There is no courses where you spend years learning before you start studying. You get 4 years worth of money or 3 years depending on the program, and that's all the time you have. So all you do is research. So I don't know. Not having studied in the US, I'm not sure how to recommend what people should do. But yeah, about the different types of programs that are available and what you want to do, I guess.
Right. There are different types of programs for sure. And I like the point that you brought up because studying internationally versus studying in the States, things can vary. But if there are any resources online that could help the students, would you mind sending that test and we could share it with them as well for what?
Yeah, if I can find some. Yeah.
Okay, great. And the last question we'll ask is more so, again, an overview of your profession. How do you overcome the challenges that you face in lab and in your work?
Try to remind yourself that it's not your fault if something doesn't work. Having a good set of friends and other people in the lab that you can talk to, work out problems with them. Sometimes even just using them as a sounding board to get through difficult things or difficult problems that you don't know how to solve. Yeah, a good supervisor that you can talk to.
That's great. Thank you so much.